Hello students. So today we are going to study the few questions based on the translation. These are the few conceptual questions which I have prepared to just give you a confidence for the topic which we have discussed in the previous video about the translation. You know very well, translation is the process by which RNA, which is bringing the coded information in the form of the sequence of nitrogenous base, is being translated in the sequence of the amino acid with the help of transfer RNA on ribosomes. Okay, this already we have discussed in the previous videos. Now let us solve the questions. So now see, first question is the process of translation will begins when? Option is given when messenger RNA will binds with transfer RNA, or messenger RNA binds with a smaller subunit of ribosome, or messenger RNA binds with larger subunit, or it binds with amino acid. We have already discussed the process of translation will begin when messenger RNA will bind with smaller subunit. Now see the next question. P and A site, two sites are present in which subunit of ribosomes? If you remember the diagram, once again recall the diagram, you will find that the answer will be coming to the larger subunit. The P site is also known as peptidyl site and A site is also known as amino acyl site. Is it clear? So your answer will go larger subunit. Next question, if you see which of the following are related or required in the amino acylation of transfer RNA. Amino acylation means activation of the transfer RNA. So for activation of transfer RNA, if you remember, we have discussed that we want the enzyme, we want amino acid, and we are using the energy. We are using energy in the form of ATP. Is it clear? So it is their first option is giving energy catalyst non-specific, wrong, it is specific. Okay, catalyst, ATP and specific, this option we can go. But let us see the third option also. Again non-specific, no, we will not take this option. GTP, no. Right now only I told you that for amino acylation of transfer RNA, we want enzyme and we want energy in the form of ATP. Now see the next question. Translation is not possible in eukaryote if suppose cap is lacking a fifth end of messenger RNA, or exons are not removed, or 5' polytail is absent, or mutation has not occurred before transcription. So by seeing this, which option will you think? First of all, this fourth option is wrong. Mutation has not occurred, that is no logic only. Actually, exons are not removed, this is true, but it is introns which are removed. Okay, and the exons are also not removed anywhere. Now, translation is not possible if cap is not there. Cap is lacking at the fifth end, then the translation will not start because it's the fifth end of the messenger RNA which binds with the ribosomal subunits. Now, next question they are giving is ribosome in prokaryotes used for translation. Ribosomes are the RNA which are acting as an enzyme, and they're asking in prokaryotes. So in prokaryotes, it is larger subunit ribosomal RNA which acts as a ribosome, which is 23S ribosomal RNA. Is it clear? Now see exit tunnel. If you remember the diagram of the previous video of translation, there I have explained it clearly that there is one exit site and one exit tunnel. Exit tunnel is an area from where the newly formed polypeptide will be coming out through larger subunit. Is to remove newly formed polypeptide. Now see the next question, the RNA which is not translated. If you know, we have discussed already the types of RNAs are many there. There is ribosomal RNA, transfer RNA, hnRNA which is processed to form messenger RNA, snRNA, small nuclear ribosomal RNA, scRNA, but only translation is done of only messenger RNA. Is it clear, class? Translation is done only of messenger RNA and remaining RNA are not translated. So I hope you can now solve directly, the answer will come fourth option. Now next question, translation occurs where? Translation occurs in the nucleoplasm or cytoplasm or cisternae of Golgi, or in all area translation occurs. So first of all, if you know, there is clear-cut compartmentalization is going on. Transcription will occur in the nucleoplasm and translation will occur in cytoplasm. Is it clear? So you have to select the cytoplasm, not nucleoplasm. Then cisternae of Golgi. Cisternae of Golgi will not do translation. It will do only packing of the translated product. So your answer will come cytoplasm. Now, charging of transfer RNA. Charging of transfer RNA means what? We say transfer RNA are charged. Are they charged when they bind with the messenger RNA, or they bind with the ribosomes, or they bind with the amino acid? What will be the answer? If you remember, we have already discussed charging of transfer RNA means transfer RNA is binding with the activated amino acid. Is it clear? After solving this, then you need to submit this. After submitting, you can review your answer. You can see back, you can go and scroll back and check it. This practice session will help you to improve. Still, if you have any doubts, then you can ask me. Thanks for watching class. If you liked it, please give it a thumbs up and subscribe to my YouTube channel.